Good afternoon. Today I'm going to present about molecular characterization of red cell enzymopathies, a tertiary center experience. Uh, so red blood cell enzymopathies are genetic disorders affecting genes which encode red blood cell enzymes, and they characteristically lead to a specific type of anemia, which is non-spherocytic hemolytic anemia. There are different enzymes which are involved in the RBC metabolisms, and the different pathways are glycolysis, HMP shunt pathway, glutathione, and nucleotide metabolism. As RBCs are mature RBCs are divided of nucleus. These are the four main enzymes which pathways which are responsible for involved in RBC metabolism. So traditionally, the diagnosis of RBC enzymopathies are measured by the specific enzyme measurement, which are followed by DNA analysis of the specific genes involved in this. So this is a list of this is the four pathways which are involved in RBC metabolism, and the ones which are highlighted in the dark red are the enzymes which are most commonly affected, and the other enzymes we can see here, glucose 6-phosphate dehydrogenase being the most common one, followed by pyruvate kinase, and followed by pyrimidine 5 nucleotidase deficiency. So what is the Indian context in case of enzymopathies? So in India, G6PD accounts to 8.5% of the 8.5% in prevalence, and PK deficiency regarding pyruvate kinase deficiency. It was seen in 3.2% of neonatal jaundice in one study. Whereas in case of GPA deficiencies, there are very few case reports, and one big large series was published by Varang and Jamal et al. Regarding the in view of this individual case reports and specific enzyme deficiencies, we try to look for what different RBC enzymopathies as an overall cohort in our tertiary center in CMC Vellore. So regarding materials and methods, this is a retrospective analysis of a total of 64 cases, out of which G6PD specific Sanger sequencing was performed in a suspected case of G6PD deficiency case, out of which there are a total of 22 of these cases. This is from 2004 to 2016 duration. And from 2017, we started performing targeted NGS panel to look for all kinds of enzymopathies and other hemolytic anemias. There are 42 of these cases, and specific enzyme levels we measured only regarding G6PD using quantitative spectrophotometry assay. This is the NGS workflow, where, as I mentioned previously, this is a clinical exome sequencing of a total of 5,200 clinically relevant genes with an effective depth of 100x, and FASTQ files were trimmed, and secondary analysis were performed on CLC workbench. Later, tertiary analysis was performed using GenoMAD alternate frequency of less than 0.1, and sequence odontology was filtered, and variants were called according to the ACMG guidelines. So this is the results. A total of 64 cases, out of which G6PD by Sanger sequencing was positive in 22. On NGS of 42 cases, 29 were G6PD positive. 6 cases are pyruvate kinase resistance, pyruvate kinase deficiency. 4 are GPA deficiency, and 2 cases are pyrimidine 5 nucleotidase deficiency, and 1 case is digenic mutations, which includes the G6PD and PKLR. So this is the summary of pathogenic variants which we have seen, out of which 80% of the cases in our cohort was G6PD deficient, 9% are pyruvate kinase deficient, 6% are GPA deficient, and 3% are pyrimidine 5 nucleotidase deficiency, and 2%, which is 1 case, has G6PD and PKLR deficiency. So regarding the sex distribution, we can see here majority of them are male. This is the mutation spectra distribution of the G6PD cases, which are specifically according to the class specific, which are mentioned here. The majority of our mutations are G6PD Varissa type, which are seen in 19 patients out of the 52, followed by G6PD Mahidol and G6PD Mediterranean. These are the novel variants. Out of these 7 variants, 4 were previously reported by Arunachalam et al. And the remaining 3 cases are novel, which were not previously published. Coming to the pyruvate kinase deficiency, the median age of our group was 4 years. 5 out of the 6 are females. And three regarding hemoglobin, it was 6.6%. 3 out of our 6 patients are neonatal hyperbilirubinemia, and one had cold agglutinin disease. Regarding GPA deficiency, the median age was 8 months, and there were a total four cases reported, and median hemoglobin was 5.3%. And this is the lollipop model, which showing the mutation distribution in the GPA. So coming to the pyrimidine 5 nucleotidase, this was seen in 2 patients, and they both are in homozygous state. So majority of our cohort is 80% is G6PD deficiency. Out of G6PD deficiency, G6PD varicella being the most common, which accounts to 19 cases. And the second most common is the pyruvate kinase, which are seen in 6 cases. Out of the 6 cases, 2 cases were reported previously by Indian population by Varangi et al. So remaining 4 cases in our cohort are novel, and it is not published previously from the such lead and open variation database. Regarding the GPA deficiency, 1 out of the 4 cases were previously reported in Indian cohort, whereas 2 cases of our pyrimidine 5 nucleotidase was not reported in previously. And so to conclude, this is the first study to best of our knowledge, which looked into the spectrum of enzyme deficiencies in hereditary non-spherocytic hemolytic anemias, and that too in Indian context. So in our cohort, G6PD is the most common, with G6PD varicella being the most common. And even though G6PD is the most common, second one, pyruvate kinase enzyme deficiency and GPA are also seen in significant proportion. So to identify the true prevalence of the different enzymopathy, different enzymes deficiencies, targeted NGS panel will help in this.